Hey guys, welcome back. This is Lei. In this video, I'm going to show you how to perform differential binding analysis and motif analysis in ChIP-seq data. Like RNA-seq data, most ChIP-seq datasets have multiple samples in different groups. So we can compare differential binding sites between groups. The tools we're going to use for differential binding analysis are basically the same tools we use for differential expression analysis, due to the fact that both peaks and genes are genomic regions, and we're just comparing the number of reads that fell into these regions. In the same way, we can identify up-regulated peaks and down-regulated peaks, and these differential peaks can then be subjected to motif analysis. Sounds easy, right? Well, here's the catch. In RNA-seq data analysis, each sample has the same set of genes, while in ChIP-seq data, each sample has different number of peaks. How do we solve this problem? We solve this problem by creating a common peak set across all the samples, and then quantify this peak set in each sample. The two and the example dataset we use here are from our previous tutorial. Here is how we do it. On the Linux system, we first activate our HOMER environment we created in the last tutorial, and then we create a directory for the merged peaks. Then we merge all the peaks in the merge peaks function from the HOMER package. The input of this function are the peak files. Here we have 4 samples, therefore 4 peak files. The output is the merged peaks. The -d given option means we only merge the overlapped peaks. Once we have the merged peaks, we can annotate these peaks using the annotatepeaks.pl function from the HOMER package. The input is our merged peaks, as well as the tag directories we created for peak calling in the last tutorial. So we have 4 samples, therefore 4 tag folders. The output is the merged, annotated, and quantified peak file. The no adjust option, that means we want the raw counts, we don't want adjusted counts. HG38 means the genome we use here. So the final peak set is in the following format. So we have the coordinates of the peak set, and we have the quantification in the last 4 columns. These are raw counts. And then we have gene information associated with the peaks in the middle, and the TSS and all other annotations. Once we have the count table for the peaks, we can perform all the downstream analysis in R on your laptop. The first step, setting up the R environment. Open your RStudio and copy everything over here to install the essential packages. And then set up your working directory. And don't forget to load the packages you just installed. Next, data formatting. The goal of this step is to generate a count table in this format and sample information table in this format. In the count table, the row names are the peak IDs. The columns are the samples. The numbers are the counts. In the sample information table, we have a sample ID column and treatment column. The R code on the left is to generate these 2 tables. You don't have to use R if you are not familiar with it. You can use Microsoft Excel to achieve the same results. Here, let me explain the R code line by line. First, let's read in our annotated peak file we generated in the previous step. Remember, we have multiple columns in this file. The whole process is just to subset this file. First step, make the first column as our row name, that's peak ID. Next, we create a peak gene meshed file for later use. Next, we extract the counts from the last 4 columns and rename the columns if it's necessary. Next, we create a sample information table manually. Sample ID, treatment, and then we name the rows by the sample IDs. And then we can save the count table and the sample information table using save function for later use. Once data formatting is done, we can perform differential binding analysis using limma. The process is exactly the same as differential expression analysis as we covered before, so I'm not going to repeat it. You can find all the code on my website ngs101.com. The results are in the same format. We have peak ID, genes associated with the peaks, log change, p-value, adjust p-value, and so on. You can ignore all other columns. You can have PC plots as before, and volcano plots showing the genes associated with up or down regulated peaks. If you don't like R, here is a shortcut for doing differential binding analysis using HOMER. We can use getDifferentialPeaks replicates.pl function from the HOMER package. The input of this function is a merged peak file from our previous step. -T to specify the tag folders of the samples from the treatment group. -B to specify the tag folders of the samples from the control group. Specify genome. The output is the differential peak file. You can also perform differential binding analysis for the samples without any replicates. That means you have one sample in the treatment group and one sample in the control group using the getDifferentialPeaks.pl function. But please don't do it. Don't do any statistical analysis without any replicates. Next, we separate our differential peaks into upregulated and downregulated peak sets for downstream analysis. The goal of this step is to generate a beta file for each peak set. A beta file is a file that contains genomic coordinates for the peaks. For example, in this beta file, there are 3 columns: chromatin, start, and end. Here is how we do it. We read into our differential peak results. Remember, this file doesn't contain genomic coordinates for the peaks. And then we read into our annotated merged peak file, and this file contains all the necessary information we need. And then we subset our up-regulated peaks using log FC greater than 1, that means full chain greater than 2, p-value less than 0.05, and then we extract the genomic coordinates of the peaks from our annotated peak file. And we do the same for the down-regulated peaks and save the beta file onto our laptop. And then we can perform pathway enrichment analysis on these beta files. Here we use our grid package. First, let's read in our beta files for up and down regulated peaks and follow this format. Submit our up peaks, get enrichment table for the up peaks, and then combine all the tables into one merged table for our enriched pathways for the up regulated peaks. We do the same thing for the down regulated peaks and then combine the results. Save the results onto our computer in this format. We have ID, goal terms, we have peak sets, and we have all the information for the enrichment analysis. We can also perform motif enrichment analysis on these beta files. Note this is not R. Now we go back to our HOMER package using the find motifs genome.pl function from the HOMER package. The input of this function is our beta file. And don't forget to specify the reference genome. The output is the output folder we just created above. You can leave all the other parameters as they are. We can do the same for the down-regulated peaks. In each output folder, you can find an HTML file where the results are stored in. Open the file with your web browser to visualize the results. All the links are clickable. Last, you can integrate ChIP-seq data and RNA-seq data if you have both. The key is to overlap DGs and genes associated with DE peaks. You can plot log fold chains of peaks and log fold chains of genes to visualize the change of gene expression and their corresponding peaks. For more details, go to my website and subscribe if you like it, and I will see you in the next video.